So, welcome everyone. My name is Alan Cook and I'm the Chair of the Board of Trustees for Action for ME, have been for some 3.5 years now. This is traditionally one of the big days of our year, and this day is no different to the previous events where we hold an event kindly hosted by Allen and Overy in their wonderful conference facilities, where we would have a conference on a specific theme followed by our AGM. The big thing is the conference and the conversation we're going to have for the majority of the day. At 3:30, we will have our AGM for a maximum of an hour. It's a bit sneaky really, because we'll try to get to many of you to stay for the AGM as we possibly can. I think it's important, it's not quite as interesting as the first chunk, but it is important that we do exercise proper governance over the money that donors give us to spend on behalf of this charity, and that trustees are properly elected and voted for and whatever, and that's all the bit that happens in the AGM. So it is an important part of the governance and control of this charity, and the more of you that can participate in that, the more proper it makes it, if you like. So please stay if you can. So our symposium today is on Severe M.E, and we have a series of outstanding speakers that you've read about, which I shall introduce in a moment. And we're focusing on that topic of those people that are severely affected by ME. We're, as a result, experimenting a bit more than usual in communicating this conference to those that are unable to attend in person. So because this gets very technical, I'm going to let Sonia explain exactly what we are doing to achieve that. One of the catches is we need to speak at this podium, otherwise we're not being filmed.
Okay, good morning everybody. As Alan said, my name is Sonia Chaudhry, I'm the Chief Executive. I'm just going to say in about a minute what we are doing today. The presentations are going to be live streamed. The only thing that's going to be in the camera are the people standing here. So you in the audience will not be filmed. The presentations will be live streamed. We have an online forum, so when you're having table discussions later on, people are able to contribute online. We've had people commenting prior to the event, and we have a live chat. So I'm actually going to be monitoring that with my laptop and picking up comments that people are feeding in through the day as well. We recognise we're not going to reach everybody. Some people are far too ill to be able to participate in any way. We understand that, but we are doing everything we possibly can to make it as accessible as possible. It may be that we have some problems during the day with the technology. We've already had an issue this morning with the audio. It worked for the BBC, but it didn't work for us this morning. I understand that it's been resolved. There may be things that come up. So for those people who are watching this via live stream, we will do everything we possibly can to make it work. And I apologise now if there are glitches. We just can't avoid that. During the breaks, we have a Vox Pops type setup. So by the escalator at the back, if you've got something to say, criticism, positive ideas, thoughts, suggestions, please come and say it. We want to capture the ideas that people have. With your permission, we will then make them available to other people, but you can come along and say something, and that could just be for us in terms of what we're going to do going forward. We've got lots of stuff going on in terms of social media. We have a graffiti board area where you can write down your thoughts and views, and we will capture those. For those people that want to lie down, to take some time out, there are mats at the back if you want to lie down, or if you've got cushions and you want to lie at the side of the room, that's absolutely fine. You're very welcome to do that. At the back on the right-hand side, it's quite a stiff door, so let us know if you want to go in there. There is a quiet room. There's a sofa in there, and there's just lots of space. So if you do need to take a break at any point during the day, then you are very welcome to use that room. And we know that there are people that will come and go during the day because they're not able to be here for the full day. That's absolutely fine as well. We want people to get the most out of the day, but equally to make sure that you're well looked after as well. Thank you.
The problem when you ask Sonia to just talk for 1 minute on a specific topic is she talks about all the things that I was going to talk about next, but never mind. I have got a couple of boring administrative bits that I do need to make sure that we cover. Can you, whilst we are going to be live from a technology perspective, can I ask you to turn your phones to silent, please, so that we don't get interrupted by telephones? There is no There is no fire alarm test planned today, but in the event of the fire alarm going off, appointed people from Allen & Overy will appear magically in yellow high-vis jackets and hard hats, and they will show you the way out, as I say. If it happens, it's for real, because no drill is planned today. You'll find in your packs there are feedback forms. It's really important to us if you could fill those in at the end of the day, because that helps us improve the quality of the events going forward. And because we're ever frugal with our money, as you leave, can you put your badges back in the box, please? Because we'll use those plastic covers next year. And I think that really covers most of the things that we need to cover in terms of housekeeping. So we're going to get the event underway, and our keynote presentation is from the gentleman to my left here, Professor Hugh Perry, who is the Professor of Experimental Neuroscience at Southampton University and also the Chair of the Neuroscience and Mental Health Board at the Medical Research Council. I hope you'll all find what he has to say really interesting. Hugh, over to you. Thank you.
Well, thank you very much, Alan, and good morning, everybody. And I thank the charity, Action for M.E, and also Sonia in particular, for inviting me to speak to you. You will notice the similarity between the colours on the slide and Sonia's dress. She thinks I chose that in order to honour her dress sense, but that's not quite true. So what I'm going to do is I'm going to tell you a story in about 40 minutes, a bit less than 40 minutes or so. And it's a complicated story about interactions between the immune system and the brain. And if you think the brain is complicated enough on its own, then deciding to draw it together with the immune system adds a degree of complexity that I think all of you will recognise is really a huge problem, raises many problems, diseases that involve the immune system and the nervous system. So to make a talk that I hope will be understandable, I hope some people won't feel patronised, and I hope that others won't feel I'm simply talking over their heads. But I hope that what is important are the ideas. There's tons of data. I could show you lots of molecules and so forth, and I'm sure you don't want to know that. What you want to know is there are ideas, and there are ideas in the field that could come together, and the idea is to make a difference. And as chair of the MRC, one of our major concerns with all the money that we spend on the Neuroscience and Mental Health Board is we want to make a difference in the end. And I hope that some of the ideas that you'll hear here will help to raise new ideas, new opportunities for making a difference. So when we think about the immune system and the nervous system, until just a few years ago even, most people, when they thought about that, they thought about diseases of the nervous system such as multiple sclerosis. And many of you will have heard of multiple sclerosis. It is believed to be an autoimmune disease in which the immune system attacks the brain. And it has a robust and very particular pathology that's well studied. The consequences of that pathology are pretty well understood, and how this then gives rise to the symptoms that people see. And this has dominated the field. This idea of inflammation in the brain was dominated by what went on in multiple sclerosis. However, in the last 5 or 10 years, there's a growing recognition that inflammation may contribute to many different diseases. So degenerative diseases of the nervous system, Alzheimer's disease, Parkinson's disease, and so forth, may contribute to psychiatric diseases, and indeed, the window grows all the time. So how do these interactions between the immune system and the nervous system take place? And I'm going to address two issues. Homeostasis, this word that we use to describe the normal state of events, and then pathology. So let's deal first with homeostasis. So all of you will know about these symptoms. Everybody, there's almost no human who will have escaped feeling these symptoms. And these symptoms we refer to as sickness behaviours, the behaviours associated with a cold, with another infection, with a bladder infection, and so forth. And we see these symptoms of sickness as something bad. And the first thing that you do is rush off to the chemist and get a non-steroidal anti-inflammatory to lower your fever and, as you hope, to take away many of these symptoms. However, a number of years ago, in fact in 1988, a veterinary scientist, Ben Hart, made this comment, a very important comment that I'll read to you, that sickness behaviours are not a maladaptive and undesirable effect of illness, but rather a highly organised strategy that is at times critical to the survival of the individual if it was living in the wild. So remember, this is a veterinary scientist. So what was he talking about? So imagine an animal living in the wild. Here he shows a dog. And this dog becomes infected with a pathogen, a bacterium, for example. And the first thing that happens in this dog is the dog develops a fever. And this fever you might think of as being a bad thing. But in fact, fever is very important because raising one's fever leads to the inhibition of growth of the pathogens, and this is true of both bacteria and also of viruses. But raising your body temperature, raising your core body temperature by just 1 degree is metabolically very expensive. It uses a lot of energy. So there have to be behavioral changes that will compensate for that increase in fever, and these behavioral changes include changes in sleep, reduction in activity, the sorts of things that you might expect what you would do to conserve energy. In addition, there's a loss of appetite, and this loss of appetite is important to prevent the organisms getting the micronutrients that it needs in order to survive. So this is now a slightly different perspective on those sickness behaviours, that in fact they're important for survival. Adopting those behaviours is a good thing. So the idea that fever is good, that fatigue is a necessary consequence of raising a fever, that the hyperalgesia and so forth might lead an organism to protect itself, all of these things begin to make sense from an evolutionary point of view, from the perspective of an animal living in the wild. We know that this is also true of humans, that when there is a systemic inflammation caused by a bug, experimental studies done by Neil Harrison at Sussex University has shown that regions of the brain become activated when he injects people with an agent to mimic aspects of an infection. So there are changes in the brain when there is an infection in the periphery. So, interesting problem. How is it that an infection in the lungs or the bladder or the guts, how does that communicate with the brain to lead to these changes? What are these pathways, these immune pathways between the periphery and the brain? So this has been reasonably well studied. Surprisingly, we don't teach this to most of our young medical students. Given it's rather important, and one of the major reasons why people go to the doctor, it always surprises me. But actually, we don't teach this routinely to our medical students. So what we know is that when you get an infection, there is an immune response. And these immune cells, the macrophages, the T cells, the white cells, they arrive at the site of infection, and their job is to kill this microorganism. But when they do this, they release a whole complex of molecules. The names are not important. Please don't think you have to remember the names. And these molecules, known generically as cytokines, circulate in the blood and then communicate with the brain. We know this is true in experimental animals because we can do many manipulations to discover what sort of molecules are involved. We can show that if we take these same molecules and we inject them in tiny doses into particular regions of the brain, we can evoke these behaviours. But do we know that's true in humans? Yes, we do. So if we take a couple of examples here, so this molecule here, alpha interferon, is given to people who have hepatitis C infection, which will of course damage the liver. So to clear this virus, people are injected with large doses of this cytokine. It kills the virus, but in 50% of the people, it causes severe depression and anxiety. So simply that molecule communicating with the brain leads to depression and anxiety. Another molecule, beta interferon, given to people with multiple sclerosis because it modifies the immune system. Many people stop using the drug because it provokes flu-like symptoms. So these same molecules that we've studied in our experimental models, when used in people, produce the same effect. So we think this circuit is true not only in laboratory animals, but also in people as well. So how do these molecules change behavior? That seems to be a bit of a gap here. Molecule in the blood, brain in a box here. And of course, this idea that the brain was isolated from the immune system has been dominant for many years. The idea that the brain lived behind a special barrier called the blood-brain barrier and was in some way isolated. We know this not to be true. So there are roots, nerve fibers from the thorax and abdominal cavity that innervate, that make connections with your lungs, with your liver, with the spleen, with the guts, and so forth. And those nerve pathways can communicate signals about inflammation into the brain. There are little bits of the brain where there is no barrier between the blood and the brain, special sites around the brain, and there some of these cytokines, as I used the term, this is where they communicate with the brain. But probably the most important pathway is this pathway here, the blood vessels of the brain. And the signaling takes place across these blood vessels. So the molecules circulate in the blood, they bind to the cells that make up the blood vessels, so-called endothelial cells, and then they talk to the immune cells in the brain. Yes, there are indeed immune cells in your brain. All of you have immune cells in your normal brain or your healthy brain, and so too in the diseased brain. So all of us have immune cells in the brain. So let me introduce you to these immune cells in the brain. And you may think that this has been known about for centuries. It hasn't. It was only widely accepted about 25 years ago that there were indeed immune cells in the brain. And they're cells of the macrophage lineage, the white cells that you find in the blood. The monocytes, when they get into tissue, are called macrophages. And when they're in the brain, we give them a special name just to confuse everybody else, like you. We call them microglia. So they're a specialized cell type found in the brain. And they enter the brain in the embryo. They have the very early arrival into the brain in the embryo, and then they stay there for life. They remain there for the whole of your life. So they may have memory of many of the things that happen to you. And we know that they play a role in the normal brain, not well defined yet, but some of the things we know about. But we also know they're very important in many pathologies. So this is interesting. They play a role in the normal brain. They also play a role in pathology. And then there are other macrophages as well, lots of other populations. I won't dwell on them. And some of these sit around the blood vessels, the so-called perivascular, around the blood vessel macrophages. But these are the important fellows, the microglia in the brain. So one of the things we know about these cells is it took immunologists and neuroscientists until 30 years ago to recognize that these were immune cells in the brain. And one of the reasons that this took so long is when an immune cell gets into the developing brain, the brain keeps it under control and switches it off to such an extent it doesn't actually look like an immune cell. It doesn't show many of the molecules that its friends in other tissues express on their surface. So this tells you something important. It tells you that the brain has evolved mechanisms to keep immune cells under control, and presumably for some good reason. Presumably it's important that these cells are tightly regulated in the context of the brain, and in a way that is quite distinct from other tissues. And if I was talking to a science audience, I'd now tell all of you how exciting each of these many molecules are and why it's really, really interesting. And I would do, if you're interested, I'll bore you all over tea or coffee or about all of these different molecules. It's very exciting science that is emerging at a fantastic rate. So when the macrophage, the microglial cell, is in the brain, it's under tight control. But one of the things we know about macrophages in all tissues of the body is that they can exist in many flavors. And their job is to defend you from infections. They have been described by my old friend and colleague, Simon Gordon, as the first line of defense against tissue injury and infection. And one of the reasons they're the first line of defense, we think about them that way, is that they have an extraordinary repertoire of different receptors on their surface to recognize different pathogens. So they recognize viruses, they recognize bacteria, they recognize parasites, they recognize these many different microorganisms that come, of course, in many different flavors. And in order to respond to those, they have to be able to mount many different types of responses. So, for example, they have to mount a bacterial killing response. And a bacterial killing response is not only important, but essential. But you can imagine, just think about it just for a minute, if you only had this bacterial killing response, and it went on and on and on, and didn't have pathways to switch it off, this would be really dangerous. So there are other flavors that the macrophage can then adopt. It responds in a bacterial killing mode, and then it has to move into a restoration mode. But you could imagine that if it was too activated, it could cause bystander tissue damage or collateral tissue damage. Kind of unfortunate word used by the military these days. But this collateral tissue damage, you want to kill the bugs, but it could lead to other damage. So it's very important. They're highly plastic cells. They're very dynamic. They're meant to switch from different states, and this will become important as we go on. And of course, in organisms such as ourselves that live for decades and may have diseases that last for decades, how these phenotypes, how the particular flavor of macrophage may change over time becomes very important. So, what I told you was these signals come into the brain, and when they arrive in the brain, they communicate with the blood vessels. They then communicate with these macrophages and microglia, and then the microglia in turn signal to the neurons, nerve cells in particular parts of the brain, and you change your behavior, amount of fever, and so forth. So that's the homeostasis, that's the normal state of affairs that should happen in people from youth to old age. And one important, very important message is that these should be transient, of course, that we mount a fever, it should be a transient fever. The other behavioral changes that come with it should be transient, and this should not be associated with any damage to the nerve cells. This would be a really bit of poor design if you designed a system where this caused tissue damage. So this is homeostasis. This helps you to return to good health. So let me now set off in a slightly different direction. And I'm going to talk about chronic neurodegenerative disease for a few minutes, and then I'm going to pull these two strands together, and you'll see why, and you'll see where we're going. So bear with me if you think, why on earth is he now talking about Alzheimer's disease? Hopefully it will become clear in a few minutes. So we know that Alzheimer's disease is now the most common form of dementia in the UK. There are some 800,000 people in the UK with dementia. And the important point here is that this is a chronic neurodegenerative disease. This is a normal, healthy brain. This is a brain from a person who died with Alzheimer's disease. And you can see there is a massive loss of neurons from the tissue. And this is caused, we believe, by these abnormal proteins in the brain. Now, Alzheimer's is but one of many types of dementia and many different types of neurodegenerative disease. But all of these neurodegenerative diseases in both humans and animals, experimental models of these diseases, have this thing in common, and that is that these microglia change from this state to this state. So the microglia are labelled green, some very clever molecular manipulations. All the microglia in this mouse are green. And you can see that they're small and they form a pretty regular pattern across the brain. But in this animal that has a brain disease, a neurodegenerative brain disease, there are clearly several important changes. One is there's a lot more of these cells. Two is that they look bigger and fatter. You don't have to be an anatomist to see that there are these big changes. And they appear to be activated. They've changed from being quiet, quiescent, held under tight control, to what we refer to as an activated state. And this is true in all of these neurodegenerative conditions, and this is what my laboratory studies. So we were interested in a really simple question. What would happen if instead of having a brain where these microglia were under tight control, kept tightly regulated, but now there were lots of them, and they had begun to be released from this inhibition that we see here? What would happen if now signals came from the periphery to say you have an infection? Would it be the same as this or different? And I can imagine if I asked you, well, of course it's going to be different. The signals are going to be bigger because there's more of these cells. And maybe more because they've changed. They're now in this activated, changed state. So to cut a long story short and 10 years of research into a few lines, we did exactly that experiment. We had animals that had a brain disease, and we asked what would happen if we gave them something to mimic a bacterial infection or a viral infection. And it had two very obvious and clear effects. The first effect was acute and obvious exaggerated sickness behavior. So instead of just having a little fever that lasts for a short period of time, they now had a much bigger fever that lasted for a long period of time. Very unusual. The reduction in activity, the reduction in doing things that are normally associated with these changes lasted longer and was more severe. And so all of these sickness symptoms were exaggerated in these animals that had this increased number of microglia in the brain. It also had some other consequences, and that was it accelerated the disease itself. So that's kind of interesting too. So maybe there's now some collateral damage. Maybe it's not so benign after all. This signal arriving in the brain, maybe it exaggerates the degeneration in the brain. And all of these studies gave rise to this rather simple-minded idea that the microglia in diseased brains and in different states might become primed. So what do we mean by primed? And this is really the idea that I'd like you to take home. So in the normal healthy brain, the microglia are under tight control. Quiescent, resting, all of the sorts of holiday adjectives that people use about these cells. In pathology, and as we'll see, caused by a number of different things, these microglia become activated. They become more, they're released from this inhibition. But then, when a signal comes from the periphery, we can change them from being relatively benign to potentially tissue damaging, and generating signals that lead to exaggerated symptoms. So they're primed by pathology or by some other event, and then when ordinary signals about infection and peripheral tissue injury and peripheral changes arrive, they now give inappropriate or maladaptive signals. For those of you in the audience who are scientists and care about these kinds of things, I could explain to you, we know a little bit about how this works. We know that a macrophage exposed to one stimulus will change, so it has more receptors on its surface, it has lots more signaling molecules inside it, and when the second signal comes along, the trigger, it now makes a bigger response. And that seems intuitively obvious. So first thing, gets it ready, second trigger makes a big response. And I think that is intuitively fairly obvious. So this may be true of mice. Was it true of humans? And so we asked, again, a very simple question. And to do this, I joined forces with Clive Holmes, who's an old-age psychiatrist who works in Southampton. And we asked, and for some reason nobody else had bothered to ask this, for more than casual observation. So every old-age psychiatrist I've ever met, and indeed many people I've met who care for people with dementia, all know that people with dementia, when they get a urinary tract infection, they have a cold, they have a fall on the stairs and hurt themselves, this makes their symptoms worse. Transiently, but it makes them worse. So is it really the case? If we were to follow it for 6 months, would we find evidence that people with inflammation also had worse symptoms? And this rather complicated looking figure, the black bars tell you about the people who had inflammation. Yes, they were more agitated, more depressed, more anxiety, more apathy in those people who had inflammation and brain disease as well, relative to those who only had the brain disease. We also found, and in this context, your interest, that this is not what we're talking about in the context of ME and CFS, a degenerative disease of the brain, but it turns out that also this is accelerating the disease in people with Alzheimer's disease. And of course, that's of great interest to us in that context. So that's all great, interesting. So microglia can be primed by the presence of a degenerative disease of the brain. And when a second stimulus comes along, they amplify that signal and generate greater layers of sickness behaviors. Could the microglia be primed by something else? Are there other conditions that might lead to this? And a number of years ago, as the date shows on the bottom, we did an experiment that at the time didn't seem to have anything to do with this. In fact, we weren't even thinking about these sorts of issues. But they were to do with viruses getting into the brain. And virus delivery of molecules by viruses into the brain is a big issue in ways of potentially treating a number of different diseases. But the important point is delivery of a virus to the brain. So what was interesting is that we had mice living in what in those days was called conventional housing, which meant that they just sort of lived in a shed, really. Actually, not quite a shed, but it wasn't as clean as a pathogen-free environment. So all animals in the UK that are used in experiments live in what is called a pathogen-free environment. They're not allowed to get coughs and colds. I mean, mice do, surprisingly, get coughs and colds and all sorts of infections that all of you get. Different viruses, so don't worry if you bump into a mouse. So they get these infections, but because it complicates life, now all of experimental work is done in pathogen-free environments. Whereas it used to be done in conditions where there might be low-grade pathogens around. Now again, you don't have to be an anatomist to see there's something different. So the brown staining tells you about the immune response in the brain of this mouse versus this mouse after the delivery of a virus. And what is clear is this is a massive response compared to what we see over here. So simply growing up in a more, if you like, immunologically activating world leads to a bigger response in the brain when a virus gets to the brain. Well, pretty interesting, particularly if you care about gene therapy and all those sorts of things, but it's a complicated experiment because we don't have a lot of control over this bit. So what if we do a better experiment? We control it better. So what we did was we had mice that were given an infection. And they were given an infection, a salmonella infection, salmonella poisoning. Hopefully none of you have had it. Common in foodstuffs. You may remember terrible business over salmonella in eggs. How many chickens had salmonella infections that did not a lot of good for Edwina Curry's political career, if you remember. Anyway, so salmonella is common infection, which is why we use it as a model. And in mice, if they get a salmonella infection, usually they clear it in 3 weeks. Doesn't make them terribly ill or anything, they just have the infection. It activates the immune system, and then the immune system kills off the bugs, and the mice are fine. But now if we want to know how did that affect the brain, we can ask what would happen if we now were to see how responsive the microglia were to an inflammatory agent. And without going into the details, again, I think it's pretty clear. This is a mouse that never had an infection, and the numbers of microglia, the brown staining, is really very modest. The mice that had an infection but have now cleared the infection, they don't still have an infection, the infection's gone away, it's very clear this is now a much bigger response. So the microglia were primed, this time, not by ongoing degeneration, but by exposure to a prior infection. So primed, and then they retain a memory, and one can demonstrate this activation state has changed. Of course, this begs an interesting question: how long might that last? So this is really the core idea I'd like you to get hold of. So this is what Hart said, that this was homeostasis, that an infection led to this change in behavior that was homeostatic. But the hypothesis now is that systemic infection and inflammation that leads to sickness behavior in an individual with a healthy brain is a homeostatic mechanism with no long-term consequences. But this is distorted and maladaptive in those with primed microglia. So once the microglia get to this primed state, the response to this signal from the periphery can now be maladaptive because it can give rise to this longer lasting, if you like, sickness behaviors or components of sickness behavior. So how are we going to get to this? How are we going to understand whether in the brains of individuals with either systemic disease of one sort or another, there is or is not activation of these microglia? And the tricky bit is, of course, to look at these microglia, we'd have to take a brain sample from you. And I can see you looking a bit nervous at the idea of giving up a brain sample. It's not great. One can't do it. So are there non-invasive ways? Are there ways of looking in the brain at the microglia without giving up a brain sample? And yes, indeed, there are now. So what has been done is individuals have, in fact, many different teams have made molecules that will bind to activated microglia. They won't bind very well to the quiescent, resting microglia in healthy brains, but if the microglia become activated, this molecule will now bind to them. So that's the first step. Second step is, if the microglia bind this molecule, can we see it? Yes, we can, by making it radioactive. And using appropriate levels of radioactivity and a PET scanner, it's possible to put somebody into a PET scanner and ask, do they have activated microglia in their brains? And this has been going on now for a number of years in many different diseases. This is an experimental study to ask whether peripheral inflammation caused by an inflammatory agent derived from a bacteria called LPS, does this activate the microglia? And what you see here is really an indicator. So as the color goes more red, it means there's more binding of this microglia activation indicator. So this molecule is binding to lots of microglia that are now activated in this brain 4 hours after the LPS, when it didn't before. So it's now possible to monitor activated microglia in the living human brain. Pretty interesting question. So now we can ask, does this happen not in an experimental condition, but does it happen in people with systemic inflammation? So we now go to people. And there's a very interesting study from Drake and colleagues at Manchester University. Now they're interested in stroke, so they were interested in why people who are obese or people who have cardiovascular vascular disease, people who smoke, drink too much and so forth are at risk for a stroke. What is it about their biology that makes them at risk for a stroke and makes the stroke likely worse? And one hypothesis would be the microglia are primed in these people before they have this vascular accident. So they took hold of some people, and these were people who obviously like watching television, eating a lot, etc., and they have raised levels of this molecule in their blood called CRP, C-reactive protein. It's an indicator of how inflamed their periphery is. And you can see here these 4 subjects across the top all have evidence of microglia activation. The radioactive molecule is binding to their microglia. And even though these people are living their unhealthy daily lives, their microglia are activated all the time. So we would suggest they're primed. And so, of course, they're more likely, or at least predisposed, to have a stroke when compared to these rather more healthy people here with low levels of inflammation. So this is an interesting question now. How widespread is this idea that microglia might be activated, that they might be primed in other conditions? And in the context of this charity's work and this meeting, a very interesting paper recently published by a group from Japan, Nakatomi and colleagues, looking for evidence of activated microglia in the brains of what they call people with CFS M.E. It's a small study. The first thing that's always important to recognize, small study, first of its kind, only 9 subjects at the present time, but very interesting observations. These are the control people down here who have some microglia activation, particularly in the brainstem, lower regions of the brain. But what's pretty clear is it's different in the people with CFS ME. So the microglia appear to be more activated in the brains of these individuals. So what they then did was a correlation, and this is a correlation. You have to be clear. Correlation does not mean causation. It does mean a correlation. It's interesting. So if they looked at the amount of activated microglia here in the amygdala and compared the cognitive scores of these people, there was a strong correlation. So poor cognitive scores are related to activation. They looked at the levels of depression relative to activation in the hippocampus. There was, again, a good correlation. And finally, too, evidence that pain was correlated with activation in a region of the thalamus that one might expect to be involved. So this is really, and I think the important point of this message here is, there are now tools to enable one to study the immune system in the brain in the living human. And that this can give rise to observations that I think are provocative and interesting and make one think about this disease state in different ways. Are there ways of modifying this state of the microglia? And if there were, how would you set about it? And so forth. So this idea might be that these microglia in these brain regions are primed, and even very low-grade infections coming from the periphery or signals from low-grade infections could be sufficient to further activate the microglia and induce the symptoms of sickness behaviors. And we now know that the molecules generated by inflammation overlap with the molecules generated by stress. So simply stress is enough to generate some of these inflammatory cytokines. The notion of CFS-ME being a disease in which there may be dysregulated immune cells in the brain might be seen by some people as being a stretch too far. But the reality is this is something that's already ongoing in pain research. There is a lot of interest in the role of microglia in chronic pain. So all of you will have experienced pain, and if one's fortunate, you stab yourself with a kitchen knife, it hurts, and with time it goes away. But in pain states, chronic pain states, the pain persists, even though the stimulus has resolved. And there is a growing body of evidence that cells that may be key in maintaining that activated circuitry, the circuitry of pain, that the microglia may be involved as well. We can think of another state where there is activation of the nervous system in the absence of a peripheral signal, and the most obvious example of that is tinnitus. If you have tinnitus, you don't have to have any sound. You can be in the mountains in the middle of nowhere, and the ringing in your ears is still there. There is no stimulus. It's driven simply by the circuits in the brain. So one has to take on board these ideas that chronic pain states, many chronic states could have, and I say could, but there's a lot of work to do to get beyond the could have an innate immune component that may play a role. And having a clear hypothesis is one way forward of dissecting out the role of these potential immune cells in your brain. So the story I've told you emerged from many different studies, lots of very talented young people working with me, colleagues in other universities, and we've been funded by these agencies over the years. Thank you for your attention, and I'd be happy to answer any questions. Thanks very much.